Hello, today I will be presenting on nucleotides synthesis as well as summarizing how ATP synthase actually generates ATP. So to overview, we will begin with the review of how glucose is broken down and how ATP is generated by ATP synthase, followed by the key product formed by the PPP, and next how UMP and IMP are formed, and then finally how nucleotides are synthesized. Note, our starting materials are glucose and carbon dioxide. So here is our 4-week-long lab project of our metabolic pathways. I will reference these numbers throughout the presentations, so keep an eye out for those. We start off with glycolysis, in which one of our starting materials, glucose, is converted to pyruvate. Pyruvate is then converted to acetyl-CoA in the pyruvate dehydrogenase complex. This acetyl-CoA can then enter the citric acid cycle, in which high-energy electron carriers such as NADH and FADH2 are formed. These high-energy electron carriers can then enter the electron transport chain to help generate ATP. Once the electrons from NADH and FADH2 are donated to the complexes of the ETC, the movement of the electrons causes protons to be pumped into the intermembrane space from the matrix of the mitochondria. This creates a gradient across the membrane. This is basically creating a proton motive force, which is the driving power of ATP synthase. It has two parts, a chemical and a charge gradient. Now, let's review the structure of ATP synthase to figure out how it works. ATP synthase is a large complex enzyme, and it consists of a FO subunit, which is in the inner mitochondrial membrane, and an F1 subunit, which protrudes inside the matrix. The F1 subunit is responsible for the catalytic activity of ATP synthase. It is composed of 5 types of polypeptide chains, including alpha, beta, gamma, delta, and epsilon. Note there are 3 alpha and 3 beta subunits, and they make up the bulk of ATP synthase and form a hexameric ring. The active sites are present in the beta subunits. The stalk portion consists of gamma and epsilon proteins, and the gamma subunit is a long helical coiled coil extending in the center of the alpha-beta hexamer. Note, each of the 3 beta subunits is different since it interacts with a different face of the gamma subunit. This will be important to understand later on. The FO subunit is hydrophobic and contains the proton channel of the complex. It is connected to F1 by the central gamma epsilon stalk and the exterior column. The 3 active beta subunit sites have 3 functions. 1. Trapping ADP and free orthophosphate ions, which is accomplished by the L or loose conformation. 2. ATP synthesis, which is done by the T or tight conformation. And 3. ATP release, which is done by the O or open conformation. The proton motive force causes the 3 sites to sequentially change functions as protons flow through the F1 subunit. ATP synthase basically has a moving or a rotor part, which is the C ring made of the gamma epsilon stalk mentioned, and then it also has a stationary part, which is just the remainder of the structure. So how does this all actually generate ATP? Well, the influx of protons will cause the rotation of the gamma subunit, which drives the interconversion of the 3 forms of beta subunits. As mentioned, the T form will synthesize ATP, but is not able to release it. So a 120-degree rotation of the gamma subunit would cause a conformational change from the T to the O form, which would help the ATP be released. This rotation would also allow additional ADP and phosphate to enter the O form once the ATP departs. Another 120-degree rotation would then trap the ADP and phosphate due to the conformational change to L. These rotations keep occurring due to the proton motive force and allows the synthase to keep generating ATP. Now that we have generated the ATP that we need, we can move on to the initial pathways needed to synthesize nucleotides. We again consider glucose as our starting molecule, and the first step where hexokinase converts glucose to glucose 6-phosphate with the input of ATP. Based on the body's needs, glucose 6-phosphate can enter the phosphate pentose pathway. The main products of interest from this pathway is NADPH, which is required in many other pathways in the body, as well as ribose 5-phosphate, which is what we will focus on. So before deep diving in, the PPP has an oxidative phase and a non-oxidative phase. In the oxidative phase, glucose 6-phosphate is converted to ribulose 5-phosphate, and in the process, the mentioned NADPH is also formed. The non-oxidative phase generates other intermediates which are important as well with the help of transketolases and transaldolases. But for the purpose of this presentation, I will focus on just the first step in the non-oxidative phase, which is the conversion of ribulose 5-phosphate to ribose 5-phosphate via enzyme phosphopentose isomerase. Ribose 5-phosphate is an important component of RNA synthesis as it is a sugar present in the molecule, and deoxyribose sugar for DNA is also derived from this. Ribose 5-phosphate is then reacted with the input of energy in the form of ATP, and the catalysis by enzyme PRPP yields PRPP. This is an important precursor for the synthesis of purines and pyrimidine nucleotides, but there are two key intermediates before their formation, and that is inositol and urdilate. Let's first focus on IMP synthesis. The first step is the displacement of pyrophosphate from PRPP with ammonia, which yields phosphoribosyl amine. From here, 9 additional steps are required to assemble the purine ring. The de novo synthesis of purine rings follows successive steps of activation via phosphorylation and followed by displacement. The final product is IMP, which is required to make ATP and GTP, which I will talk about on the next slide. But it is important to note that the synthesis of purines depends on tetrahydrofolate. Let's first discuss how ATP is formed in this pathway. First, adenylosuccinate is formed from IMP with the addition of aspartate. Then, AMP is formed from the release of fumarate. Last, with the input of 2-ATP and the help of kinase enzymes, AMP can be converted to ATP. Next, let's talk about GTP. Xanthylate is made from IMP with an oxidizing agent NAD+, and the addition of water. Next, with the displacement of the carbonyl oxygen atom by NH2, which is sourced from glutamine hydrolysis, we form GMP. Lastly, with the input of 2-ATP and the help of kinase enzymes, GMP can be converted to GTP. So ATP and GTP are required for RNA synthesis, but these can easily be converted into deoxyribose equivalents directly from the ribonucleotides formed in this pathway, and then these would end up being used for DNA synthesis. So that's how our purine nucleotides are synthesized. Now, moving on to how our pyrimidines are made. First, we need to make urdilate, and to do this, first, CO2, which is another starting molecule that was mentioned, is reacted with ammonia at low temperatures to form carbamaic acid. And this is a precursor in this synthesis of orotate. Carbamaic acid is first phosphorylated to form carbamyl phosphate, and then with the addition of aspartate, the reaction yields carbamyl aspartate, and the key enzyme here is aspartate transcarbamylase. Next, cyclization of this intermediate yields dihydroorotate. And then lastly, with the help of an oxidizing agent NAD+, the reaction finally yields orotate. At this stage, orotate combines with PRPP to form orotidylate. It does so through the removal of the carboxylic group catalyzed by orotidylate decarboxylase, which yields the desired uridylate. UMP is then required for the synthesis of UTP, CTP, and TTP, which I will talk about on the next slide. So how are UTP, TTP, and CTP formed? UMP is converted to UTP by the input of 2-ATP in the catalysis done by kinases. The same UTP is then subsequently used to form CTP. Basically, the carbonyl group is replaced by an amino group, and this process requires the input of ATP, and the amino group source comes from the hydrolysis of glutamine. Making TTP is a little bit more complex. First, the UDP intermediate is used to form DUDP via enzyme ribonucleotide reductase. This enzyme helps go from ribonucleotides to deoxyribonucleotides. Then, from DUDP, we will lose a phosphate to form DUMP. Tetrahydrofolate is then required in conjunction with the catalysis done by thymidylate kinase to form DTMP. And then, again, with the help of kinases and the input of ATP, we can finally form TTP. Note, TTP is required only for DNA synthesis. And again, similar to before, CTP ribonucleotide is transformed into dCTP for DNA synthesis through similar pathways mentioned. So finally, all the nucleotides we need are made. For DNA base pairing, it is adenine to thymine and guanine to cytosine. The nucleotides needed are DATP, DTTP, DGTP, and DCTP, formed from the ribonucleotides. During DNA replication, the base pairs are matched by DNA polymerases and a phosphodiester bridge is formed, which causes the release of pyrophosphate and yields 2 ions of orthophosphate in the process. And for RNA, the base pairing is adenine to uracil instead of thymine, and then guanine to cytosine. The nucleotides needed are ATP, UTP, GTP, and CTP. During RNA synthesis, the base pairs are matched by RNA polymerases using a template DNA strand. And again, a phosphodiester bridge is formed, which causes a release of pyrophosphate and yields 2 ions of orthophosphate in the process. So that is how ATP synthase works and provides the ATP needed for DNA replication and RNA synthesis, and also how other nucleotides are formed as well. Thank you for listening.